Hello guys, I am Shubham from Tutorials. This video is about transposition. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. Till now we have completed the site-specific recombination topic. So now this video is about transposition and the examples of this transposition we will be discussing in the next video. First of all, what is transposition? Okay, so these elements which can move from one position to another. From the name only, we can understand the position is changed. Okay, so these are also known as mobile genetic elements. Mobile genetic elements means what? Mobile means what? Which can move. So those elements which can move from one position to another. Okay, move from one position to another. So this is transposition. Let's see with the help of diagrams. So see here, this is a old site, transposon. Okay, the element which can move from one place to another is known as transposon. Now what happens? This is a new site and this is the old site. Now in here, there are two types. First of all, movement without duplication and movement with duplication. So what happens here? In the first type, just the position is changed. Okay, just this will go in this side, right? And in the second position, second type, what happens? It sit here also in the old set also, as well as in the new site because of this duplication. We know what is duplication. We can say this is known as cut and paste. You might heard the transfer of files in the laptop is done by these two mechanisms: copy and paste and cut and paste, right? So in the first part, it is cut and paste. Means what? From the old site, it is getting deleted, and in the new site it is getting inserted, right? And the second type, it is copy and paste. Okay, copy and paste means it will remain in that side also as well as in the new side. Okay, so what is responsible for this? Duplication is responsible, right? We know duplication is what? In duplication, same segments are repeated, right? Suppose this is a chromosome, right? And here are the two segments. This will get duplicated like this. Okay, so this is known as duplication. First, only two copies were there. Now there are four copies of the same segment, right? This is duplication. So in the first step, duplication does not occur. Cut and paste mechanism. In the second step, duplication takes place and the mechanism is copy and paste, right? Now, see, these are the five different species, right? Maize, humans, Drosophila, then yeast, Saccharomyces cerevisiae, and Escherichia coli, bacteria. So this green, what we can say in the green is the transposable elements. Okay, green color is transposable element. Transposable element means those will give rise to transposition, right? So in maize we can see high number, very high number. Humans also have, we can say not much, but less than maize. Drosophila is having less than humans, right? So like this, the order is decreasing of transposable elements. Okay, so here order is decreasing. We can say more number in maize and least number is in bacteria, Escherichia coli. Okay, there are no transposable elements. Only we can say the regions, two regions, which are green color. Alright, this is just a what we can say overview of the transposable elements found in different species. Now these are three classes of transposable elements. Three classes. So first is what? DNA transposon. Okay, so these elements we have to know. Transposase. Transposase is what? Enzyme that bring about transposition. Okay, so transposase, or sometimes it is called integrase as well. Okay, so transposase, this element is coding for transposase. These are the terminal inverted repeats. We can see terminal inverted repeats means the sequence will be like this. And here it is target site duplication. This is target site. Same arrows pointing this direction. Right, means what? These are direct repeats. Okay, these are inverted repeats and these are direct repeats. Right, so these DNA transposons have a simple structure, we can say, but the next type, virus like retrotransposons or retroviruses. Okay, retroviruses, these have LTR. See, LTR, long terminal repeats. Okay, long terminal repeats are there, as well as in the center integrase and reverse transcriptase enzyme will be coded. Okay, integrase and reverse transcriptase. What is reverse transcriptase? RT. First of all, it is responsible for reverse transcription. We know what is transcription. DNA to RNA. Okay, DNA to RNA. I will write here DNA to RNA. This is known as transcription. Right? This is very basic. Now from RNA to DNA. RNA to DNA is called reverse transcription. Reverse means what? Ulta. Right? Reverting the process of transcription. So this is a reverse process of transcription. DNA to RNA, making DNA from RNA. Right? So here these are the segments LTR, integrase reverse transcriptase, as well as same target site duplication, terminal inverted repeats are also there here. Okay. And in the C, the third part, poly A retrotransposon. These are like mRNA thing. See, poly adenylation is there, tail. Okay, poly A tail. Then here it is 5 prime UTR. What is UTR? 5 prime and 3 prime UTR is there. UTR is a untranslated region. Okay, so this don't give rise to proteins. Okay, so these are proteins which are not formed. Okay, because these are untranslated regions. So translation does not occur here, right? And this ORF and ORF2, these are very important segments. What does ORF1 do? ORF encodes RNA binding enzyme. RNA binding enzyme, right? And ORF2 encodes enzyme having activities. I will write here enzyme having activity reverse transcriptase and endonuclease. So both of these activities are present. Okay, so this is enzyme 1, this is enzyme 2. So these two enzymes are produced. ORF1, RNA binding enzyme, and ORF2, enzyme having reverse transcriptase and endonuclease activities. Right? Now let's see the mechanism. First of all, DNA transposons. Okay. Okay, so this is the first mechanism. Cut and paste. Cut and paste transposition. Okay, DNA transposition by cut and paste mechanism. Right. So here, see, we can see here these are the terminal inverted repeats, and this is a DNA element. Right. Now what happens? This will encode an enzyme transposes. Okay, and that transposes will bind here. See, forming a synaptic complex. Okay, what it forms? Synaptic complex. Then just we know cleavage will take place. DNA cleavage of both the strands. See, cleavage takes place, and then this will attack the target DNA. See, we can see here this 3' OH and this 3' OH will attack the target DNA, right? Then what will happen? DNA strand transfer. Just reactions will take place. This will go and bind here, right? The DNA repair synthesis to fill the gaps, basically joining the strands. Okay, so this gaps has to be filled. So here we can see element in new DNA location and the ligation of NICs completed. This is a new DNA. See, this is the new DNA synthesized, and this is by target site duplication. That's it. Before going to second mechanism, see here are the three types of cleavage actually. So cleavage takes place by the three mechanisms. Let's see what is those. Okay, three types of cleavage. Now see, this is a transposase cleavage. Here is the gap, here is the gap, means here cleavage has took place. The first mechanism TN7 is there. So these are the enzymes TN7, TN105, the Hermes. Or okay, so here what happens now? TNSA comes and cleaves here. Okay, just here what happens in the second thing? This end will attack here, as well as this will attack here. Okay, so 3' OH ends are attacking the opposite strand, right? So your transesterification reaction take place and your hairpin intermediate is formed. This is known as hairpin. Okay, and then again the hairpin will open. We can see here hairpin will open and then this will attack the target strand, right? Now in the third step, what happens? Again, hairpin formation only takes place, but here, see, here, loop formation takes place. This OH is attacking opposite strand. This OH is attacking opposite strand. That's it. And after the breaks are occurred, it will attack the target DNA. Right? Got it? Very easy. Just the three types of cleavage are discussed here with the help of diagrams. We can see here this is the hairpin formation here, and here the hairpin formation take place here and here. Right? Now the second type of transposition is the replicative transposition. Okay, this is replicative transposition. Okay, replicative transposition. From the name only we can see that replication will be taking place here, right? This is a donor DNA, you can see here, and this is a transposable element, right? Now this attacks a 3' OH after cleavage attacks the target DNA, we can see here. Okay, then strand transfer takes place. So this will transfer the strand here. Replication fork assembly. From the name only we understood replication will take place, right? Replicative transposition. So replication fork will assemble here, and then replication will take place through the transposon. We can see here lagging strand, leading strand, all the replication things we have studied previously. Continued replication, ligation, right? Then ligation will take place. Means what? Joining of strands. And this is known as co-integrate. Very important word, co-integrate. Means what? The two strands or two DNA are integrated, joined to each other with two copies of transposon. Okay, we can see here this is a transposon and this is a replicated DNA. Again, this is a transposon, this is a replicated DNA, right? So this is about transposition, DNA transposition. Two types. First is cut and paste, and the second is replicative transposition. Then now this is the second type of transposition, retroviruses. Okay, retrovirus, virus like retrotransposons. Here what happens? Reverse transcription. See, first of all, transcription gives rise to this RNA, right? After RNA reverse transcription cDNA formation take place. Okay, this is cDNA. Then integrase catalyze 3' and cleavage. 3' and cleavage by integrase. Right? See. Then this is the 3' and cleavage. Then this will attack the other strands. Can see here from the image. Same here. Nothing is different, only the starting mechanisms are different. After the cleavage, target DNA is attacked and then integrase catalyzed and DNA strand transfer and here ligation. And this is a new integrated copy. See this. Okay, target site duplication. Alright, this takes place by the replication, new integrated copy after ligation. After ligation, this is the copy form. This is about retrovirus. That's it. Only here important is reverse transcription. Reverse transcription. Okay. First of all, DNA to RNA, then RNA to again DNA. Then 3' OH formation. And that 3' OH will attack the target DNA. That's it. Then the last thing is the poly retrotransposons. Okay, this is what? Poly A retrotransposons. Okay, so here little line DNA. Okay, line sign is the example of poly retrotransposons. So what happens here? Line DNA is there, right? As well as transcription takes place, RNA formation. Okay, line mRNA. Then line mRNA, we know here there are two segments, ORF1, ORF2. Then here formation ORF1 and ORF2 proteins are produced. What they will do? Bind to line mRNA. Okay, they will bind to line mRNA. Then what will happen? This will go and attack the target DNA. So this is the target DNA. Here the ORF with the mRNA is attacking. Then what happened? Target site cleavage. RNA DNA hybrid formation. Right? RNA DNA hybrid formation. Cleavage will take place and the synthesis of cDNA strand. See, this is similar to the retroviruses or retrotransposon. Okay, line cDNA is being synthesized. Then what happens? RNA degradation and second strand transfer. Okay, second strand synthesis. Then DNA joining and repair. And repair of what? For the proper ligation. Okay, proper joining will takes place and the synthesis of line DNA has completed. What it means, we started from the line DNA and in the end also line DNA is being synthesized by this process, poly retrotransposons. Okay, poly A retrotransposons. How? Reverse transcription. Okay, so first of all, RNA will be formed. This is RNA. From RNA, proteins, ORF1 and ORF2 proteins. These proteins, and from this RNA, this will go and bind to the target site. Then there will be formation of the cDNA, complementary DNA. And then after completion, the DNA will be joined and the line DNA is inserted. That's it. So easy it is, right? Similar to the processes. Okay, one more thing of this transposon is that one more thing. The transposons are divided into two types. Which are those? First of all, autonomous. Autonomous and second is non-autonomous. Okay, now in autonomous what happens? Autonomous elements have the everything. Means what? They can do their own transposition. Right? They have everything required for the transposition process. In non-autonomous, the transposes is absent. Transposes coding element is absent. Non-autonomous requires help of the autonomous to complete the transposition, right? Because transposes is not synthesized by the non-autonomous. Okay, transposes gene is absent, and here it is present. So it will take transposes gene from autonomous. Okay, I will write here autonomous. It can do by own. Okay, transposition. And here in non-autonomous, it requires help of autonomous. Okay, so that's it for this video. This is all about transposition. I hope this video helps you. If you find this video helpful, do let us know in the comment section.